That was our cue. Hi, everyone. Good morning. My name is Lima. I work at Scripps Institution of Oceanography as a postdoc. I'll be the moderator for today's session. We have 7 different talks, and to make sure everybody has enough time, we're doing them in lightning style, so it's 5 minutes for each presenter, followed by 1 question from the audience, and we're going to have a 2-minute time slot for each presenter to address that question. So I encourage you to definitely ask your question, but also if you couldn't ask your question during the session, follow up with the speakers afterwards. Scientists, we love talking about our projects, so encourage you to reach out. Our first presenter is Jennifer Burnaford she's a professor at California State University, Fullerton, and working on multi-agency rocky intertidal network. So welcome, Jennifer Burnaford.
All right, hi, thank you. So I'm going to give you a very, very brief introduction to a large-scale collaborative network that does work monitoring rocky shores from Baja all the way up through Alaska. All right, so the Multi-Agency Rocky Intertidal Network has a mission to provide data that are publicly available and useful for managers at the state, local, or federal level to get information that allows the long-term assessment of rocky shores and provides baseline data in case of either slow changes in the rocky intertidal zone or dramatic changes like from an oil spill or similar event. So you can check us out on Instagram, and our webpage is a great resource because, as I'll talk about later, our data are all publicly accessible and downloadable from our website. So the network was started officially in the early 1990s based on work that had been done by the National Park Service at the Channel Islands, their efforts to monitor their rocky shores. And then in 1997, they created the Marine Network officially. And this was detailed in a recent book chapter. And the really key thing about the network is that it involves multiple groups, and every group comes in with their own funding. And so it's a sort of a tapestry of funding and effort by different groups that have allowed us to have continuous monitoring of rocky shores since the early 1990s. So collectively, Marine has data from over 300 locations along the entire Pacific coast. And we have a number of different data types, which are indicated at the bottom: biodiversity monitoring, community science, temperature monitoring, sites that we have gone to after an oil spill. And you can look at all of our data on our website, and I'm going to talk about a couple of types that are most interesting, hopefully, to people that love seaweed. So our long-term monitoring core protocols are designed to give us a really strong ability to detect changes over time at one site, and then also to compare across sites. So we have fixed plots, rectangular plots, in this case in an endocladia zone. And we go out annually to these sites and we collect data on percent cover. So at 100 points in the rectangle, we record what's at the surface and then what's underneath the understory. And we have 20 groups that do this along the coast, and we use a coordinated set of protocols as established in our protocol booklet and our handbook. And so new people coming in make sure that they use protocols that we have all been using for years. And every year we have 2 meetings to ensure that we are all consistently using these same protocols. So We have 16 teams from 20 different groups. We have 173 sites at which we collect these data annually. And these sites have been monitored for quite a long period of time. So we have 141 of those sites have over 10 years of continuous data, and 41 of those sites have over 30 years of continuous data. And all of our data are placed into a single consolidated, centralized data network, data bank. And so we upload our data into this one data bank, and we have QA/QC managers, so quality control managers, that look at all of our data that is submitted by every group, and that makes sure that along the entire way from data collection through data assessment, we are able to make sure that we have the same types of protocols. So I'm going to very quickly now go through another type of monitoring, which is biodiversity monitoring, where we have a small crew that travels along the sites at 203 sites. And these require high levels of expertise. So we have lines that go from high to low intertidal zone. We have data that we collect at 100 points along each line. We look at what's underneath our point, and then we go outward and we look at the next 2 species that are localized to our point. And that way we get not only percent cover estimate for a site, but also a species list for the site. We enter our data into a consolidated app, a specialized app that allows us to also identify whether things are on boulder or cobble, whether they're epiphytes or attached to the surface. If we can't identify a species in the field, I have 30 seconds, we either phone a friend. We sometimes call experts on FaceTime. So these are museum persons. We take our algae back to our hotels or in the at parking lots on the side of the field. We press them. We create voucher specimens that we then identify off time, or we take them to the algal expert in California, Dr. Kathy Ann Miller, and we have little algae parties every year to make sure that we are correctly identifying all of our species. Our data, are used by Cal OSPR, and that's the Office of Spill Protection and Response, and MPA assessments. Our data are available to the public, so if you go to our website, you can download data from any of our sites. This is from Point Fermin, which is right around the corner. We have long-term data and biodiversity data available to you and available to our managers. So if you want to follow us, pacificrockyintertidal.org, and we're also on Instagram. Thank you.
We have time for a quick question.
Yeah, I was just curious, since you're looking at things on rock, if you consider the nature of the rock, if anybody's addressed, say whether it's granite or Miocene, shale, whatever.
So we do have, for all of our biodiversity sites, we have rock specimens. We actually collect pieces of rock with permits. And so we have records of the different types of rock at the different sites. And we do have that information if people wanted to evaluate the data based on rock type. So that is part of the data that we collect. And then we, for whoever wants the data or the surveys, they can take that into account when they do the analysis. And I'm around all day, including giving a poster in the poster session, so you can all ask questions later as well.
Thank you, Jennifer. Okay, our next speaker is Frej Tulin. He's a postdoctoral fellow at Carnegie Science, studying brown algae responses to light signals. So let's welcome Frej Tulin.
Good morning, everyone. Yeah, my name is Frej. And today I'll give you sort of a brief intro to our work to understand the sort of basic biology of photosensing and the response of brown algae to different light signals, what we can call photo development. So yeah, brown algae, they live in a very dynamic environment. one dynamic parameter is light. Right? So light can change a lot in intensity, basically how bright it is with depth, for example. Also, the color or the quality of light can change depending on where the algae grows. And so why are we interested in understanding how the algae responds to these different colors of light? Well, it's well known that plants and algae can sense different colors, and that determines in many ways how they grow and when they decide to reproduce, for example. And that is important for understanding sort of their ecology in the natural environment. But it's also, and perhaps more importantly, relevant for sort of rational approaches to aquaculture, where a mechanistic sort of insight into the light response, the light responses can lead to, say, greater control of the reproductive cycle in, let's say, a hatchery setting, and maybe improved biobanking, storage, and retrieval. And it might also provide targets for breeding of light-dependent traits, as some examples. The organism that we used for these studies is a small brown algae called Ectocarpus. And it's a great model system for this purpose because, first of all, it has very well-defined and quantifiable phenotypes or responses to light that we can measure. It also has efficient CRISPR gene editing protocols so that we can delete individual genes and look at the function of those genes in response to light. And also has a fast life cycle so that we can perform genetic analyses at a reasonable timescale. This slide here shows the main developmental process that are relevant for this talk. The algae starts as a single spore, about 10 micrometers in size, and then it germinates and grows into this prostrate filament that is attached to the dish, the culture dish in this case. And you can see it has these kind of round cells in the middle that are differentiated and photosynthetically active. And then it goes through a transition where it grows an upright filament into the water. And this is the one that's going to produce reproductive spores of various kinds. So that's an important transition. Now, we discovered that when you germinate these spores in the complete absence of blue light, so they're grown under a decent amount of monochromatic red light, then you lose these typical morphology of the filament. Specifically, in this case, you can see that they lack the rounded cells. Okay, so they don't develop normally and they grow very slowly. A very small amount of blue light fully restores the normal development of the initial filament. This is 0.1 micromole photons of light per meter square in seconds. So this is small. It's a small amount of light. So this means that this weak blue light acts as a signal to activate photosynthesis and growth in Ectocarpus. Now, it's also interesting that if you maintain the algae under this low blue light, they keep growing and they grow very well, but they remain vegetative. Okay, so what you're looking at here is kind of a tuft of vegetative filaments that is kind of sticking to the surface. If you crank up the blue light to 2 micromoles, there's still a low amount of blue light, but a little bit more, then you get robust production of these reproductive filaments. So, and we can quantify the amount of growth in different colors of light and different amount of blue light sort of added, and the sort of amount of reproductive filaments that they produce. For the purpose of this talk, so this leads us to the model where basically we think that there may be 2 receptors a very sensitive receptor for low light and a slightly less sensitive receptor for the higher light, and that they drive different developmental processes. And with that, I'll thank you all for listening. This is the great team that I get to work with, and our sort of ongoing work is focusing on these questions right now. Thank you.
Thank you, Frej. We have time for one question.
Have you looked at other wavelengths of light?
No, we not really. We've looked at green light also. The green light, we don't have a strong phenotypic response with other wavelengths of light. So the reproductive decisions is mainly triggered by blue light, but they have photoreceptors that respond to red and green light, but we don't know what they do yet. Okay.
All right. Our next speaker is Lauren Smith, postdoc researcher at University of California, Santa Barbara, working at the intersection of aquaculture, conservation, and climate change. Welcome, Lauren Smith.
Hello. Normally I'm trying to convince my audience to love seaweeds as much as I do. It feels like a real easy lift in this crowd. So today I'm going to talk about rockweeds. These are a foundational species. They're canopy forming. They're found in the mid to high intertidal zone, and they provide really important shelter for invertebrates and are associated with higher algal and invertebrate diversity. These gorgeous guys up here are the three common ones you can find in California. There's Fucus discus, Pelvetiopsis californica, and Sylvicia compressa. Unfortunately, rockweeds are in decline globally. And this is due to a number of different issues: pollution, water quality, and a suite of climate stressors. In California, Sylvicia compressa is declining. That's this one here, the golden rockweed. Look how gorgeous she is. This has led to a decrease in invertebrate diversity, richness, and abundance in Southern California, where the declines are occurring. The threats to Sylvicia and other rockweeds are kind of interesting because they live at the interface between land and sea, and so they are faced with both marine and terrestrial stressors. And in particular, it seems like Santa Ana winds are a big stressor for Sylvicia compressa, and that can lead to high winds, high temperatures, and low humidity. So we know that I work in an aquaculture lab, and so we know that there are some aquaculture practices that we could apply to restoration that might help increase our output. And so we're starting to culture rockweeds in the lab. So we collect these guys here. So these are the receptacles, the apical tips that have all of the reproductive material. And we bring them back to the lab and put them on these settlement discs. And we stress them enough that they open up their insides where they keep their eggs and sperm and drop those down on the settlement discs. Those then grow into rockweeds. And so that's not like a fossil, that's just an imprint from the receptacle of where all of the baby rockweeds fell. Once we have baby rockweeds, they go in UV treated water. It's a closed system, so there's hopefully no bacteria coming in. We use industry standard feed, a 12-12 light and dark cycle, and we have set up pumps so that we have 2 high and 2 low cycles a day to mimic their natural habitat. So what if we made it even better? And we're going to do that through priming, hopefully. So priming is when you expose an organism to a mild stressor. The memory of that prepares the organism for future stress conditions. So here you can see all of our plates inside of our closed mesocosm. So this is a low tide. And we have added little fans and reptile heaters. And the key is you stress them just enough, but not too much that they die. So we're doing a priming stress first. So about half of our mesocosms get low levels of both the fan and the turtle light for 3 hours a day during the low tide for 3 days. They get a 2-day recovery period. And then we stressed all of them with high levels of both the fan and the lamp at low tide during the daytime low tide cycle for 6 hours a day for 5 days. So did priming work? We took a bunch of measurements that we're looking at now. We actually just finished this experiment last Monday. So we have relative growth, photosynthetic capacity, the change in surface area over time, the final dry weight. We're going to look at surface area to dry weight ratio and average community height. And just taking all the pictures and taking all the measurements to do this took my colleagues, there was 2 of us or 3 of us, about 8 hours just to collect all this data. And so stay tuned. Here I have a picture at 4.5 weeks of growth. So that's before priming and then after priming. And you can see it's starting to fill in. We are seeing differences, but we'll have to keep you posted on if the priming worked. Thank you everyone for listening. And I want to thank my funders and collaborators at the Nature Conservancy, the Frej Lab, and UCSB. And I'm going to plug the Restoration and Seaweed Solutions panel with my colleague, Dr. Erica Nielsen, which is at 2:15 today in the courtyard. And I'm happy to take any questions.
Never really knew that they had like this sort of memory. Is
Yeah.
it that when they're put under stress, they develop in a different way, so the next time they have stress, they already have something to prevent it more?
Kind of. So I think it's epigenetic pathways that are doing this. And so I think that can be, and this is not my area of expertise, like through histone modification or there's one other that I'm missing. But yeah, so the idea is if you have experienced it before, then you might have expression that is different because of that experience, and especially if it is at certain stages of your life cycle that might be able to carry on to the future. And some are short-term and some are long-term. I have time for one more.
Other than rockweed, has anyone done heat testing or heat stressing with any other species?
Yes.
Local intertidal species of algae?
I don't know about local intertidal species of algae. So it's pretty widely used in agriculture, and we've done it with kelp. So at very juvenile stages of Macrocystis spirifera. And there have been some other folks that have done rockweeds, but I think more with subtidal stressors, so more marine stressors than terrestrial stressors, is my understanding at this time. It's kind of new for macrophytes. Great, thank you.
Okay, our next speaker is May Blundell. PhD candidate at UC Davis, uses modeling and simulations to study kelp forest intervention strategies. Welcome, May.
Thank you very much. So today I'll be sharing some relatively new results from my modeling work simulating kelp enhancement strategies for bull kelp and giant kelp. So as we get through the talk today, I'll first outline what are some current challenges and questions regarding kelp forest restoration. I'll really highlight modeling as a tool to explore the complexities and the range of decisions that need to be made. And then I'll get into one result that highlights the importance of outplanting the microscopic life stages. So many of you probably know this story. 2 species of canopy-forming kelps along the coast of California in 2014 through 2016. 2 events, marine heat wave, die-off in sea star populations, and these led to more than 90% loss of bull kelp and more than 50% loss of giant kelp along the California coast. And impacts lots of things, including fisheries and the kelp-connected community members along the coast. So what are some current challenges and questions regarding kelp restoration? Well, first, kelp forests are naturally variable. They naturally increase and decrease over time. So how do people know when is they're a real signal or cue of decline that signals a real issue that needs intervention. Next, what is the threshold that we're looking for before we intervene? How bad does it have to get before managers and restoration practitioners decide to invest resources? And next, because of the natural variability, there's potentially opportunities to save on resources by waiting for natural recovery. And then we must also think about how much intervention is required. That's like, how many kilograms do we put out into the water? And what's the duration of this intervention? And then this next point about life stage. So the kelp recovery might depend on the buffering provided by different life stages, like the little baby microscopic stages or the adult stage. And so here are 3 different life stages that all have undergone or are undergoing trials by different restoration practitioner groups in California. So the microscopic life stage, then what if you grow them out a little bit more in the lab and outplant them as juveniles? And then the next is drift kelp supplementation. So putting out pieces of kelp to try to suppress the grazers because they like the drift much more than they prefer the drift over the live kelp, and they'll eat it when they have it. Okay, so modeling is really a tool to explore all those different complexities. And here is how my model works. I follow the life cycle of kelps. So here we are in the early fall, late summer, when it's adults that are in the water. About now in the fall, they are dropping their spores. The spores settle onto the sea floor. They develop into these microscopic gametophytes. I combine these spores and gametophyte stages into an M microscopic stage. Also important in the winter is these storms that break up the kelps and produce a lot of drift, and that deters the grazers from eating the live kelp. And then in the spring, they're growing up again as juveniles. So a model is like a computer game. I make the rules and I let the computer play it. And here's what the computer gives us for bull kelp in a historic climate scenario with low grazing pressure. So you can see that most of the years are neutral years. That's the 2s. And then it's interspersed with some bad years, like marine heat waves, and some good years, like strong upwelling. And you can see in the biomass that the kelp forest dips down in bad years, comes back up in good years. And so if I repeat the simulation 100 times and summarize over those 100 simulations, I get some summary statistics that tell me how this kelp forest is doing. So how do I actually model restoration? I say, okay, computer, if a cue of decline, that's either the juvenile recruits in the spring or the adult biomass or some signal of grazing pressure, if the cue goes below some percentile threshold, then we're going to wait some number of years before choosing which life stages to outplant and then how much effort to go. So here's a result from those simulations. On the horizontal axis, I'm showing you how much effort is going into the outplanting in terms of kilograms. And then on the vertical axis is the outcome. So what's the adult biomass outcome that we get from this restoration activity? And I'm coloring these points by the life stages that were outplanted. And you can see that more effort, you get more output, but it saturates after some point. And really highlighting that it's those brighter colors where you have the M, the microscopic outplants, that you really get a result. So takeaways, modeling is useful, outplant the microscopic stages, and let's think about how to enhance the survival of those stages. And you can catch me at Western Society of Naturalists to learn more. Last little bit, I want to plug a cool art project that I did, music and dance, inspired by this work. And if you're interested, please pick up a program from me throughout the day. Is it okay that I leave this box on the stage for the rest of the presentations or session? Okay, so programs will be in this box right here, and please pick them up if you're interested. Okay.
Hi. Thank you. May has time for one more question.
Yes.
Hi, thanks so much.
Can you tell us a little bit about how your model, or whether your model uses like cause of decline, agent of decline, whether it's temperature, grazer, and how that informs, when and what restoration methods to employ if the goal is to bring the kelp back?
Yeah, so the question is about how am I getting those year type effects into the model? Like how am I actually?
Yeah, like sometimes it's hard to determine why kelp isn't somewhere where it used to be, like how does that play into the model?
Yeah, so I have those good years and bad years, and the good years are inspired by upwelling of the nutrient-rich water. Bad years are inspired by marine heat waves. And so the model works by generating a random sequence of those year types. And the frequency of those year types, how they come up, is inspired by climate projections from the IPCC. And then how I'm actually getting those effects onto the kelp is I'm knocking down the reproduction, the recruitment, the growth. I'm knocking the good things down in bad years, and I'm boosting the good things in good years. And that's how that happens. And I'm reading papers, combing the literature, trying to get a sense of, okay, how much does this knock need to be to get a realistic-ish result? Does that answer your question? Yeah. Okay.
Okay, our next speaker is Yuanzi Wu, postdoctoral research associate at Hubbs-SeaWorld Research Institute, working on seaweed and invertebrate restorative aquaculture. Welcome to Yuanzi.
Hello, everyone. So my name is Yuanzi Hu. I come from Hubbs-SeaWorld Research Institute in San Diego. So I will introduce you my seaweed aquaculture research in integrated aquaculture systems. So first of all, I want to introduce the concept of the IMTA, integrated multi-trophic aquaculture. So this picture shows the single species aquaculture. So we feed them 100% feed, but nearly 75% of nitrogen and 77% of the phosphate will go into the environment. So accumulate the nitrogen and phosphate will cause severe ecosystem consequences, such as the eutrophication, harmful algal blooms. So if this waste can be eaten by other trophic levels, for example, the shellfish, the seaweed, so the farmers can produce more seafood production and make more money without any additional cost. So this is the concept of the IMTA. You see here is the fed aquaculture fin phase. Feed the feed, produce a lot of fishes, and the nutrients can used by shellfish and the seaweed, also the sea cucumber deposit feeders. So this is the concept. IMTA can produce the additional seafood without additional cost. So seaweed play vital roles in IMTA systems. You see, this is a fish cases. We culture the fin fish, abalone, sea urchin, and shrimp. They produce a lot of fishes, solid waste. And they also produce a lot of nutrients, also can produce oxygen lower and a lower pH. But if we co-culture seaweed, it can produce a lot of biomass of seaweed, and clean the seawater can go back to the fin fish tank, all can discharge into the coast. And then this seaweed can produce the food for human and can produce the feed for animals, and a lot of product can produce. So I will introduce my research on seaweed in IMTA system. This is our two species cascade IMTA, very simple systems. So you can see the water, the effluence from the raceway of the water sea bass can go into the seaweed tank by gravity. So we can co-culture water sea bass together with other seaweed species. So we optimize the conditions to maximize the seafood production and nutritional quality and the nutrient waste removal efficiency in this system. This is multi-species cascade MTA system. We can co-culture water sea bass, sea cucumbers, and sea urchins. You see, all animals co-culture produce the nutrients can uptake by seaweed. This is so the water flow direction from here to here in water-based tank and the seaweed tank. We can use the produce the seaweed to feed the sea urchins directly. This show a little bit culture, our temple culture tank for the ova and for the dolls. This is our recirculating amine system. You see here is the water recirculating direction from the seaweed tank go to into the fin-fish tank and back to seaweed tank. So clean seawater can go back to the fin-fish tank. Also, the solid waste can backwashed by drum filter to mussels, sea cucumbers, water clean back to seaweed tank. You hear from the seaweed can uptake all the nutrients produced by co-cultured animals. So seaweed have high nutritional quality and can remove a lot of nutrient waste. So summary, we summarize our system productivity for the ova, glycerol, and DOS. So kind of the productivity, here's the protein content, also the uptake rate on nitrogen and carbon. So it's a pretty good system. So next step, if we can do, I want to do a large scale culture land-based seaweed. This is I get from this video from a colleague in China. Also, we have net pen in Carlsbad and Santa Catalina Island. So we want to set up the open sea IMT systems. Thank you so much.
Like, will it fall apart really quickly without maintenance, or is it quite robust and self-sustaining?
You said the maintenance, right?
Yeah, like, will it self-sustain itself? Like, will it be able to, like, fix itself if there's issues, or will it start to fall apart really quickly if, like, say, something goes wrong?
Yeah, the land-based IMD system, we need to set up the series tank from here to here to here. So for our two stages, for example, just a fin-fish and seaweed. So you don't need to do a lot of, but you need to do the same thing with the fishes from the fin-fish tank, the general maintenance. But for seaweed, you don't just harvest per week. Depend on the size of the tank. But for the multi-species, because we need to just maintain a tank for the sea cucumber species, but we don't need a lot of time. For example, you could culture abalone together with the sea cucumber, but normally you need to clean the tank every week for the abalone. But the culture with the sea cucumber maybe 2 weeks, so can reduce time. Thank you.
Our next speaker is Phillip Tahimic, master's candidate at Cal Poly Humboldt, working on the CLASH system. It stands for Correlate Apparatus for Seaweed Hatcheries. Welcome, Phillip.
All right. Hello, everyone. Thank you for being here. I'm Philip with the Department of Fisheries Biology. My graduate advisor is Dr. Rafael Cuevas-Uribe, who's with us here in the audience today. So, as mentioned, what is CLASH? Correlate Apparatus for Seaweed Hatcheries, modular seaweed nurseries from Moss Landing Marine Labs with a central lighting component designed to give homogeneous conditions throughout the system. And this is suitable for micro and macro algae. Here on the right is how our system appears at Humboldt's Marine Lab. Coupled with this infrastructure, we're doing a little technique called gametophyte culturing. Why are we interested in kelp gametophytes? The idea here is we have more control over planting and harvesting times. If using traditional methods of seeding from spore, a farmer would need to go and collect this reproductive tissue in the summer or fall. This is called a sorus, and do this for every growing season. Alternatively, a farmer can set up their own gametophyte cultures after just harvesting every few years. And once established, these provide a source of seed stock that is available independently of the spawning season. Another reason we'd want to use gametophytes is greatly reduce contamination in our systems. This is a picture of our nursery and some old clash system growing media when we seeded with gametophytes. And here's what that system looked like just 1 year prior when we seeded from spore. We had severe green algal contamination, which competes with the kelp for light, space, and nutrients. With that in mind, the objectives for this project were to, of course, set up the gametophyte bank, see the clash system with a representative kelp. We went with Alaria marginata, winged kelp, and we want to develop a protocol from the nursery stage to grow out in tumble culture, as the clash project is designed to be repeatable and open source. We started off with our gametophyte bank. Red lighting keeps the individuals vegetatively growing and keep them at the gametophyte life stage. Due to their low metabolic rate, they can remain in conditions of low light and nutrients. And once we had amassed enough biomass through this process, we moved on to seeding. We used unglazed ceramic tile to mimic a natural substrate. Applied our gametophytes with either paintbrush or spray bottle and slotted them into 3 of our clash systems configured in a closed system. Here is a trend of the tile cover after every week, reaching about 90% kelp cover at the time of harvest. Here's what that looks like visually between our 2 inoculation methods. There was no difference in cover or harvest length between them, so either is suitable. As it happens, the best growth was not on our tiles, but on individuals that happened to settle directly on the light source, which received more light, were at a higher temperature, and they were more directly in the path of our aeration. We also wanted to demonstrate that our individuals would survive after being transferred to a secondary system. So we transferred them to a flow-through system configured as you see here. We ran it for 3 weeks and we saw no loss in the pigmentation of our winged kelp and minimal tearing. That being said, there were still some improvements from the very first trial that was conducted in the clash system. So there were some trade-offs here, and the next steps would be to tinker with the conditions here to optimize yields in the clash system and scale up the process. But that is a project for another day. I'd like to thank NOAA National Sea Grant for funding the clash system project. And please visit our booth to see a working clash system at the California Sea Grant booth on the Great Lawn. Thank you for your time. Any questions? Yes.
So you mentioned that there were issues with like the system and particularly with the seaweed accumulating in the light source. Are there any other interesting, I guess, setbacks in terms of how you're going to improve the system in the incoming future?
Yes, so in terms of setbacks, while we had a larger length on that spore trial on the previous slide, we had a lot more contamination when seeding from spore. So I think if we use the like a longer photo period that was set for the spore trial, along with higher nutrients, we can get more consistent growth and but also be able to do it year round using our gametophyte cultures. Does that answer your question? All right. We have time for one more quick one. Yes.
What type of nutrients were you using for kind of keeping the cultures going in the crash systems?
Yes, we were using closed systems, so we used half strength F/2 medium or F/4.
At what frequency?
At half strength, that would have been 0.25 milliliters of media per liter of seawater.
Thank you.
This is our final talk. Thank you so much, everyone, for coming and staying here. We're going to hear from Evan Simpson. He's a master's candidate at Cal Poly Humboldt as well, working on cultivation of Alaria marginata in Humboldt Bay. Welcome, Evan.
Thank you very much for that introduction. Yeah, my name is Evan Simpson. I'm also in Dr. Rafael Cuevas-Uribe's lab. We're going to be talking about wing kelp today. So, to get big picture, this is a map of the globe, and all these percentages around these different countries are their production of seaweed as of 2024. And we can see from this red rectangle here, the United States accounts for less than 0.02% of the seaweed produced as of 2024. So because of that, we wanted to ask the question, how can we lower the barrier of entry for farmers, specifically in the Humboldt Bay, to be able to grow Alaria marginata? So with that, we have three main objectives. The first objective is to compare growth for seasonality and yield biomass between longline systems. So we did a summer-fall grow out, we did a winter grow out, and then we also did a spring-summer grow out. We also wanted to determine the optimal growth depth for the Alaria marginata in Humboldt Bay. So the bottom system that you see right here has this basically decrease in the depth, starting at a much shallower depth and then going down to a deeper depth at about 16 feet at the bottom. And then we are also comparing the yield and quantity of Alaria marginata between our longline systems. So this top one we have here is our single longline system, and then the bottom one that we have here is our multi-longline system. The main difference between the two is that the multi-longline system has 5 lines running parallel to each other, allowing hypothetically for more growth of the kelp to be in a smaller area. But we wanted to see if that was actually going to be the case. So first thing we have to do is we have to collect the Alaria marginata. We collect from this site called Baker's Beach. It's quite beautiful. And what we're looking for is this sporophyll tissue. This is located at the base of the Alaria marginata near the holdfast. Once we collect the tissue, what we're going to do is we're going to clean and rinse the sporophyll, put it in an incubator overnight at 10 degrees Celsius. Hopefully at that point, it'll want to release. After a release, we can count the spores using a hemocytometer. And then eventually we will then inoculate either spools or petri dishes for gametophyte banking, like Phil talked about earlier. After that, with our seeded spools, we will put them into our recirculating nursery. Our recirculating nursery allows for us to control pretty much all of the conditions that we have, such as temperature, nutrient, light intensity, etc. And then hopefully after about 6 weeks, we can see some really nice growth. These are some really good spools. These ones are seeded from gametophyte. And just to reiterate, on the left that you can see here, we have an unseeded spool, and then on the right is a spool that had been growing in our nursery for about 6 to 7 weeks. And then if we really zoom in at a microscopic level, we can see that all the little kelplings are growing on the string. Pretty cute. Next thing we're going to do is we're going to outplant. So we usually start pretty early in the day. It is an all-day event. We'll get out on our farm. This is what the farm looks like on a really nice day, real calm. We'll string the spools onto our grow line. And then as we are moving the boat down, what we'll do is we'll unspool the string and attach it either zip ties or by tying it directly to the line. So some good results. Winter is best. There was no growth in any of the other seasons, and I'll get to that a little bit later. But in the one season that we did have growth in winter, we had individuals that were over 3 meters in length. The total weight of both of the single lines was about 159.6 pounds, with over 3,500 individuals counted from the lines. And we can see that, some of these individuals are pretty beefy. So probably what the reason was for this lack of growth in the other seasons that we looked at was a combination of temperature. So we can see that from about mid-October to about June is really optimal times to grow. Research has shown that Alaria marginata does not grow very well past 17 degrees Celsius. So, anytime during the summer from June to October, you're probably not going to get much growth if you outplant in Humboldt Bay. With that, salinity might be something to look at, as well as the nutrient composition of the bay. Oftentimes during the summer months, there's not much nutrients. If we look at our kelp growth over time by 10-meter transects, main thing that I want you all to notice is this 0 to 10, and then the 10 to, and then the 90 to 100. These are at either ends of the grow line, and we can see that there is not much growth in any of the other sections of the line. And this is probably due to there being a sag in the middle of the line, and then closer to the buoys where the water, or where the line is higher in the water column, there's more growth. So probably turbidity within the grow season is something that is dictating how much kelp is growing on the long line. And this was corroborated by the amount of kelp that we were seeing on our multi-line sections. So this is our multi-line, and just to kind of reiterate, the shallower part right here, so the 0 to 10 meters is where we saw all of the kelp growth. And finally, when we look at the yields between our two systems, we can see that the single line grew more than double the amount as the multi-line, thus showing us that the single line is probably more optimal for growing Alaria marginata. This may be due to abrasion, as well as the competition for space between the individuals in the multi-line, just because they're so close together. And yeah, with that, I'd like to thank my committee members, lab mates, Fish 499 students, volunteers, industry professionals, university staff, funding sources such as the CSU ARI and Dell SkiSick. Thank you.
Hi, everyone. After we take questions for Evan, actually stay here. We have one more talk. There was a blip in the system. Yeah, so I encourage everybody to stay after Evan take his question.
Any questions? Yes.
Why were you using the nodule?
We were using Curomone plant. Okay. Yeah.
I was wondering, so you said on the multi-line, you saw less growth than the single line, but
Yes.
was it the multi-line one, the one that you had angled, so you had less growth when it was at deeper depths?
Yes, but when I put that into account with this graph, because I'm only comparing the first 10 meters, the first 10 meters were pretty similar between both of the lines, because we saw that sag in the single line. Yeah, just within those first 10 meters, we saw there's a pretty large difference. Yeah. Yes.
Because I think about bull kelp a lot,
Yeah.
I was surprised that winter was a great season for growth.
Yeah.
Does that line up with Alaria's natural, how it does things when it's just living out there?
Surprisingly not, no. A lot of the individuals that will collect are more intertidal along kind of the exposed coastline, whereas Humboldt Bay is much more protected, which makes it kind of an optimal area to be growing, just because it's easy access. But a lot of the times we'll see that the adult Alaria sporophyll individuals will be senescing at around October to November, but that's when like the best time to start growing up in the bay is, so which is interesting. Yeah.
All right, now we're going to hear from Dan Abbott from Reef Check, talking about 20 years of monitoring kelp forests.
All right, thank you. So yeah, my name is Dan Abbott. I'm the Kelp Forest Program Director for the Reef Check Foundation. And yeah, we are all about this, these beautiful kelp forest ecosystems. They're incredible scientifically, like incredibly interesting and dynamic systems. They're incredible economically. They're incredibly just from the amount of life and biodiversity. And of course, areas of incredible beauty and someplace that a lot of us have a very personal connection to. So at Reef Check, we started in, but some of these ecosystems have obviously gone through some major changes recently. So our work is around monitoring these ecosystems. And so starting in 2006, we started a kelp forest monitoring program to look at these areas. You know, kelp tends to be an underappreciated ecosystem. Not everybody has access to it, and even scuba divers, like the one population that is uniquely positioned to experience these ecosystems firsthand, doesn't always, have a very solid connection to it. I mean, if you think about some of these kelps, like say Pterogophora californica, like giant, very abundant kelp, grows to 3 meters plus tall, abundant all up and down the coast from Baja to British Columbia, Alaska. Doesn't really have a widely accepted common name, right? Like folks just don't have that connection to it. So we have our monitoring program. We think monitoring is an important aspect of conservation. You can't conserve or protect these ecosystems unless you know what's going on with them. And so to do this, we utilized citizen science, so volunteer recreational divers to go out there and survey these kelp forests. This has a couple benefits. I mean, we're collecting high quality data. We are also doing it, fairly cheaply, volunteer divers. But we're also introducing people to a better understanding of these ecosystems and having them develop a connection with it. So for us, our monitoring protocol is fairly robust. We look at about 80 species of fish, invertebrates, macroalgae. We take a bunch of substrate measurements, what's growing there, what kind of rock it is. The training is very intensive. It's about 5 weeks of online. They get a stack of flashcards, a couple inches thick, that they got to learn not just species identification, but all of our various protocols. We do a pool dive, a bunch of ocean dives, and a bunch of field evaluations. In fact, the whole class is a series of evaluations. And most people pass some of the class, but they're not always able to collect data in all fields, in all areas. For us, one survey is a whole bunch of different transects. So it's kind of an in-depth looks, about 18 transects, looking at those 80 species. It takes about, say, 12 to 16 people, one dive to complete one of these surveys. So it's quite a bit of work. We started in California 20 years ago, but we have since expanded. We have a program down in Baja, all the way up into Washington, and even a little bit of work in California. We completed our 2000th survey this year, our 20th year. And on Saturday, we are scheduled to complete our 200th survey of this year. And I think overall, we'll probably get around 250 surveys done this year. Now, obviously, 20 years of data is hard to summarize in a 5-minute talk, but a couple things that we have been a part of studying and raising awareness of. Obviously, there's a lot of issues facing kelp forest up in Washington and Puget Sound. There's sort of been a steady decline of kelp, not always for reasons that are understood. Why is kelp here and not over here? So using our data and looking at different oceanographic, temperature, but also nutrients and abiotic factors. We've tried to start to understand why kelp has been in decline in this area. Obviously, Oregon and Northern California, dramatic loss in kelp. All these things can be really obvious from the surface, but not always from but you don't always know the rationale. Like up in Washington, most of it doesn't seem to be driven by urchins, where Oregon and Northern California definitely is, especially with bull kelp. Central California, also a loss, upwards towards 70%, but maybe for slightly different reasons, or why is there more here than Oregon and kelp? And Southern California has obviously had persistent barrens as well. We've also seen some invasive algae like Sargassum horneri or hornweed spreading. And of course, when we talk about kelp forests, it's easy just to focus on the kelp, right, and the forest, but there's all these impacts to all these other species. That's why we have that 80 species list of looking at what the knockdown effects are. It's enough to make you sad sometimes. But we're also looking at solutions. So starting in 2020, we started a restoration program. It actually wasn't driven by us. It wasn't driven by Reef Check, but it was driven by some of the volunteers that we had trained and had developed those connections with those ecosystems. So these divers went out, they cleared an area about 1 hectare, and kelp returned. I actually dove there 2 days ago. It's since kind of swapped back, but definitely there's a huge community component to that work. And we also launched a Dive into Science program because our effort is to get people involved in this work and involved in these ecosystems. And we understand that there's a lot of barriers to scuba diving. And so we have a program designed to get under-resourced communities more involved, teach them scuba diving, teach them scientific protocols, and have them engage. And the goal is that we can, help protect and conserve these ecosystems and turn them back to these beautiful, scientifically robust, economically important ecosystems we all love. For that, I'll take any questions. One question. Anyone? Yes.
Presentation, Dan. I was wondering if you're collaborating with any agencies who are using your data for monitoring data.
Yeah, so our data is all publicly available. It's been used by the MPA team at CDFW, OPC. All our data goes through a rigorous, QA/QC process, and we just have a form on our website where you can request data, and we generally answer about or send out our data, export it like 2 to 3 times per week. So there's definitely a lot of interest in this data. You can also go on our website and look at a list of reports and publications that utilize the Reef Check data set. So yeah, I highly recommend going on there if you want to peruse it and to fill out that form and export it and chug away. All right, thank you.
Thank you, everybody. That was a great session. Thank you to the speakers. Yeah, it's a wrap. I encourage you to stay for the next events here in the panels, outside, the lawn. Have a good day.